Samantha got her Bachelor's of Science in Biology from Western Michigan University, and her research talk is on evaluation of fish predators and biological control agents to manage and eradicate red swamp invasions in Michigan. Samantha, the floor is yours.
Are you seeing my screen? The note screen? Okay, good.
No,
all right. Good. All right, so thank you for the introduction. Before I get into my evaluation of predator species as biological control, I kind of wanted to give a brief background. So red swamp crayfish are native to the southern US. In Louisiana, they're used commercially as food. This species is also highly invasive with the ability to tolerate a broad range of abiotic conditions. And as such, they have successfully invaded over 5 continents. They also have adverse ecological effects. With native crayfish species, they tend to outcompete. This crayfish species gets very large and they are aggressive. They also dig burrows that can cause erosion. They dig their burrows right along the banks. They also tend to consume large amounts of macrophytes, which in turn can affect water quality. And so, of course, these are very a problem. And unfortunately, in the summer of 2017, the DNR first got their confirmed reports of them in Michigan, specifically in the Farmington Hills and Novi area with some in Sunset Lake. They infested ponds, both retention and residential, inland lakes, and golf courses. So because of this, the DNR and MSU teamed up to form a response plan. The initial goals were to evaluate early detection and evaluate and implement control methods. Some of the control methods that were looked at were trapping, chemical control, specifically CO2, and biological control. However, from the start, we knew that no one method would be effective for management. Specifically for biological control, this was a mystery. We needed to determine which fish species were most likely to be successful biological control agents. In Michigan, it was unclear which fish predators were likely to consume enough crayfish to be effective, and then also how these predators would go about consuming these crayfish. So there was really no data to inform DNR stocking efforts. Also, we need to take into consideration, the abiotic conditions in which these species would be stocked, again, mostly in ponds and lakes in southern Michigan, and of course on golf courses. Species that were picked also needed to be easily obtained and regulated by management agencies. Taking all this into consideration, I picked 3 species for evaluation. Those include largemouth bass, bluegill, and green sunfish. So once I picked these fish, there were still things to take into consideration, such as mouth or gape size. Each of these predators have different size mouths, and we needed to be able to determine the size selectivity of these fish to understand the range of crayfish sizes that these predators are capable of consuming. And then I also needed to develop an analytical framework to evaluate size-specific consumption. This leads me to the optimal foraging theory, which initially states that there is an optimal prey size that exists for fish predators. So through reading and literature about interactions between crayfish and fish, crayfish size plays a major role in predation. So then from that, I used the optimal foraging framework to evaluate the effectiveness of this species and also to determine the size, the selectivity and the size ranges of my fish predators. To do this, I needed to quantify these results. So I created a cost-benefit model. Essentially what a cost-benefit model is, there's always costs that are associated when predators are foraging for prey. In the case of fish, it is pursuit and handling time, the time it takes to pursue an individual, to how long it holds the individual before consumption. So here's an example of a cost-benefit optimal foraging curve. In this study, Stein created this one as he looked at smallmouth bass feeding on Orconectes propinquus, which is another crayfish species. So on the x-axis, he looked at the size of crayfish taken into consideration the carapace length. So carapace length is the measure from the tip of the rostrum to the end of the abdomen. On the y-axis, he also used handling time, or he measured the ratio of handling time pursuit over digested organic material. In my case, I looked at crayfish mass. And so essentially what these curves are, as you minimize the ratio between the costs and the benefit, you'll end up at a point where there's an optimal size prey that these fish species likes to consume. Another way to put that is if the costs are low and the benefits are high, you will start moving your way down the graph to the optimal size of crayfish. For this case, these smallmouth bass were consuming sizes that were 18 millimeters in carapace length. So armed with this information, this framework, and a way to quantify my results, I am sharing with you some of the questions that I used to pinpoint which combinations of predators would help with control. One was, does predator species affect size-specific handling time of crayfish? I looked at what size crayfish might we expect predators to consume in nature. So with the creation of the cost and benefit curves, I should be able to see differences in the ranges of sizes of crayfish that these predators would consume. And then finally, do predators differ in size-specific consumption rates? So for my methods, my objectives were to develop the optimal foraging curves and quantify size selectivity on crayfish by these fish predators. And then doing so in lab, I ran randomized feeding trials with replicates of different crayfish sizes to a suite of my predator species. There was a control for the sizes of these fish. For largemouth bass, it was 10 to 12 inches. For green sunfish, it was 6 to 8, and the same for bluegill as well. I did this to control for the influence that predator size has on the results. So my data collection consisted of, I'm going to break it down into costs and benefits. So the costs would be the pursuit time, the handling time, the benefits. I measured crayfish mass in grams. I also recorded the carapace length for each individual crayfish species, as well as the sex. So when feeding these, when doing these trials, there was a predation sequence. So after introduction, the fish would orientate themselves towards the crayfish. They would then attack. That attack would either lead into consumption or rejection. To measure their pursuit time, I took the difference between the time of orientation to the time of attack. And then for handling time, I took the difference between attack to consumption, which in my case, the consumption would cease or the timing would stop once the operculum stops moving on my fish species. So then analyzed all my data, I put it together, gathered my information. And so then here are some of the results that I have. So for handling time, I log transformed the handling time to form a linear regression. So on the x-axis is carapace length with the natural log of handling time. So as you can see, as carapace length increases, handling time increases for each of my predator species. So there was a significant relationship there between handling time and carapace length. Then for pursuit, I just used linear regression. Unfortunately, for bluegill and largemouth bass, there was no significant relationship between the pursuit time and carapace length for the species. However, green sunfish hybrids did have a significant relationship, but they had a low R-squared. Then I calculate the cost, which is the pursuit and handling time. I also transformed this data, and then there was a significant relationship between the cost and carapace length. As carapace length increases, the cost increases across species. Next, I created my cost benefit curves and my optimal foraging curves for each of my species. I used the low S smoothing on crayfish captured for each of my individual fish. So what this graph is showing, all the blue dots represent the successful captures of crayfish. The red, represents those ones that escaped consumption or were rejected. So for for bluegill, you can see that the nice optimal size for bluegill is around 14 millimeters of carapace length. For green sunfish, they kind of have a larger range, but it's somewhere between like 20 and 30 millimeters carapace length. And then for largemouth bass, it's the same. However, for largemouth bass, they were clearly able to consume the widest range of crayfish sizes compared to bluegill and green sunfish. Next, I looked at the probability of consumption. With this, I used logistic regression to characterize the probability of consumption based on the crayfish sizes for all of my species. I fit this model based on AIC values. So carapace length on the x-axis, probability percent consumed on the y. And in this case, the probability of consumption decreased with increasing carapace length for all of my species. However, the rate of decrease and the probability of consumption clearly varied between species. Overall, largemouth bass were able to consume or had a higher probability of consuming crayfish of all sizes. I thought it was very interesting that for bluegill, up to about 14%, 15 millimeters carapace length, they were more successful at consuming these size crayfish than green sunfish. However, that quickly dropped to zero around 40 millimeters. And maybe that has to do with the gape size of these bluegill. So in summary, largemouth bass, based on my optimal foraging curves, you can see that they consumed the largest range of crayfish sizes, and they were more efficient at consuming them. And then although bluegill consumed the smallest range, they are more effective at consuming juvenile crayfish. And then with green sunfish kind of in the wash, being able to consume a wide range, but still having that limitation on larger crayfish sizes. So in conclusion, all of the species that I tested were able to consume red swamp crayfish. I controlled for the limited size range of the predators. So for future investigations, we need to look at the diets of fish found in environments where these crayfish infestations are to see if they're actually consuming these crayfish, and if they're doing so kind of at the rate and within the sizes that I predicted or that I showed within my lab study. And then because control is most effective when species that are implement lower overall population sizes of these invasive species, stocking a combination of predator species is best and would ensure consumption of all or a multiple life stages of this invasive species. So with that, I would like to acknowledge the MDNR for my funding, my advisor, Brian Roth, my friend and technician, Aaron Sullivan, who I would have been completely lost with on multiple occasions without his help running all these trials, to Kelly, Sarah, and Doug, who graciously stepped in to help supervise my techs when I was collecting and putting together all my data. And then Sarah Thomas from the DNR, as well as her DNR techs, for helping to collect my crayfish for this study. And with that, any questions?
Thank you very much, Sam, for that very interesting presentation on fish predators and red swamp crayfish. So does anybody have a question for Sam? You can raise your hand or write it in the chat. Let me see if I can write in this. I cannot see any hand up. Oh, yes, Erin. Okay, Erin has a hand up. Yes, Erin, please, if you could unmute yourself.
Sure. Hi there. And thanks, Sam. That was a really cool talk. And I may have missed it, but I think you mentioned gape limitation in the beginning.
Yes.
And then I think I only caught size of predators in going through your results. I was wondering, like I said, I might have just missed it, but did you look at gape size of all of these different predators, and how did that come into play?
So I kind of used the sizes of my fish predator species to control for kind of any difference in gape size. So like I think I said that I used 10 to 12 inches for largemouth bass, and like the same for those species. I didn't necessarily measure the gapes of each individual predator, but with the reduction on the size, I was hoping that that would kind of take care of any greater variation in what they were able to consume.
Gotcha. Thank you.
All right, thank you. So next up, is it Mary? Your hand is raised.
Yes, thank you.
Thank you.
Hi, Sam. Nice work. I was wondering, do you have a sense for the growth rates of the crayfish, to have any sort of estimate for how long does a crayfish typically spend? I'm sure it would be temperature dependent and such, but how long would a crayfish be at those most vulnerable body sizes, if you will?
So
once the crayfish grow larger, then the hope of controlling them with the predators is pretty slight, right?
So that was one of the things that I wanted to look at. So not only did I want to know what kind of sizes they preferred, I also want to know the range of crayfish sizes that they were able to consume. So just because they preferred for bluegill like 14 millimeters, they were able to consume multiple sizes of crayfish, and then the goal was to see if these other predators, these other species, would kind of fill in the gaps. So if one species couldn't consume, like say for bluegill, the largest one that I was able to feed them was 20 millimeters of carapace length, 25. The bluegill, or sorry, the green sunfish and the bass were definitely able to consume those other sizes. So multiple stocking of these predators would be best, so that you can kind of, sorry, again, buffer for which ones can't be eaten by a certain predator. Does that answer your question?
Yeah, that's great. Thank you.
Yep.
Sam, there's a question from Michael, and his question is, have predators been used to control red swamp crayfish in other locations?
Yes, so in Europe, they've used the European eel to control populations of red swamp crayfish. There's huge, over there again, the infestation is fairly large. They've also used catfish. I can't remember the name of the catfish right now off the top of my head, the scientific name, but they've used catfish as well in Europe.
Okay, great. Jean?
Yeah, thanks. Great talk and really well presented. I was kind of surprised to see the difference between the bluegill and the green sunfish of similar sizes, but that was pretty interesting. So 2 questions for you. One, do you think that you would have seen a different kind of a curve with maybe a different minimum if you had looked, if you'd been able to look at like the energy or nutrient content of the crayfish rather than just relying on the mass?
Yeah.
And then my second, oh, sorry, I was just gonna say, my second question was, did you ever do a taste test?
Okay, so first, no, I have not done a taste test of resin crayfish. Where these crayfish have been coming out of, I would not want to touch any of those near my mouth. So for the other question, I'm sorry, could you repeat that again? The first one was
I'm wondering if you think you would have seen different results if you had been able to use energy or nutrient information of the crayfish rather than just relying on the body weight as their benefit to the fish.
Yes, I would have liked to be able to kind of get more of those, that type of data, but I do not think that it would vary much. I know that Stein did his project on the digestible organic material, but still, like minus like the exoskeleton and all of that. But I don't think that it would have changed it too much. But it's definitely something that I would like to investigate when I have more time to figure out all the energy contents of specific like red swam crayfish.
Great, thank you. Nice job.
Sam, thank you very much for the presentation.